The novel coronavirus, or SARS-CoV-2, has literally forced our world to come to a screeching halt. What is this virus, and why is it having such an enormous effect on our lives? Coronaviruses are a large family of RNA viruses. They get their name from their distinct crown of spiky proteins attached to their outer envelope. They use these spikes to attach to human cells and infect them, and usually causes only mild to moderate upper respiratory tract illnesses, like the common cold. However, 3 new coronaviruses have emerged from animal reservoirs over the past 2 decades that cause serious and widespread illness and death. The 2003 SARS-CoV jumped from bats to civets to humans, causing severe acute respiratory syndrome, a serious form of viral pneumonia. In 2012, the MERS-CoV jumped from dromedary camels to humans in the Middle East, causing large numbers of fatalities but remained mostly local. The latest to emerge is what we call SARS-CoV-2. It causes an illness, COVID-19, and has been declared a global pandemic by the World Health Organization since March 11th this year. Of the close to 8,000 cases in 2003, SARS claimed approximately 800 lives. MERS had a smaller reach than SARS, but a higher fatality rate. In just 5 months, over 5 million cases of COVID-19 have been reported worldwide, with more than 320,000 people dead. Compared to the previous 2 outbreaks, COVID-19 is turning out to be much more contagious, and also more than just a respiratory disease. Damage has been reported to lungs, heart, the GI tract, kidneys, brain, the central nervous system, even COVID toes. COVID-19 seems to trigger inflammatory reactions and blood clots, causing organ failures. This has prompted a reassessment of the disease and how to treat it. There are 2 broad categories of tests being developed by scientists: diagnostic tests designed to test for an active infection in the body, antibody tests to measure the immune response of the body. The most common diagnostic test in the market is to detect infection in patients is a genetic technique called RT-PCR, or reverse transcriptase polymerase chain reaction. The SARS-CoV-2 virus is known to reside in the upper respiratory tract. This requires pushing a special swab deep inside the nose or throat into the space that connects the two called pharynx. These naso- or oropharyngeal swab samples are carried in tubes containing a universal transport medium from the collection sites to the labs, which may or may not be close to each other. Once the samples reach the testing lab, it requires trained technicians on specialized machines to process them. The collected sample swab contains a mixture of human cells, virus particles, and other microbes. Sample prep isolates virus particles from the other contaminants on the swab. The extremely small quantities of viral RNA in the collected sample are not sufficient for direct detection. PCR uses repeated cycles of heating and cooling to make many copies of targeted sections, or genes, that distinguish SARS-CoV-2 from other viruses. About 20 to 40 cycles, or approximately 2 hours, are needed to amplify the viral code to detectable levels. Most large PCR machines can run the process on multiple samples in parallel. Though processing time on PCR machines is only a couple of hours, delays due to sample transit, sample prep, documentation and reporting, and backlogs due to lab capacity can all cause longer turnaround times in getting back results. Another type of test that we hear of is the antibody test. This is a blood test, also called immunoassay or serologic test. This test works by identifying the presence or absence of antibodies. What are antibodies? When our body encounters SARS-CoV-2, it mounts an immune response to it. This presents itself as protein particles, called immunoglobulins, in our blood. Immunoglobulins bind to the corona spikes, disabling the virus. It is important to note that the antibody tests should not be considered a replacement for the PCR-based active infection tests. Swab tests have the highest probability of detection right around and after the onset of symptoms. It takes our immune system some time to produce a response. This lag between onset of infection and the antibody buildup is the reason why each test serves a different purpose. While the race is on for antibodies to stop SARS-CoV-2, the science around it is still in its infancy and not yet reliable. If you've already had COVID-19 and lived through it, your body may know how to fight off a reinfection in the future, for a period of time at least. You might even be able to donate convalescent plasma to critically ill immunocompromised patients. Antibody research will also contribute towards developing effective vaccines. While antibody tests are critical to measure the pandemic's reach and long-term disease surveillance, more immediately, with COVID-19 raging across the world, diagnostic tests remain the #1 need to contain the spread of disease and protect our frontline army of healthcare and essential workers. Unfettered access to rapid, reliable, and safe tests of all types will be needed to get our stalled world back on its feet, running again.